These tiny balls, resembling lizard eggs, are actually mini-brains created in a lab that can learn anything with just a snap. Literally, scientists have crafted them, exposing them to 240 variations of 8 voices. In a mere 2 days of training, they could identify almost 80% of those 240 voices accurately. Imagine, even if you're not good at math, but you aspire to have a genius-level intellect like Einstein. Now, with these fast-learning mini-brains, this can become a reality. You just need to install these mini-brains into your body, calibrate them, and then train them for a few days to become a math genius. Can AI really do all this, right? So, has the time finally come when anyone can instantly acquire talents through AI's assistance? In this video, we'll discuss how this entire technology works, how these mini-brains gather information, how learning occurs, and ultimately, how this new mini-brain can be integrated into our minds. But before moving forward, there's a challenge. Have you noticed it? Our brains already process a considerable amount of information, enabling us to effortlessly perform daily activities. Now, if we dedicate a part of our brain solely to a specific task, considering the limited size of our skull, some compromises might be necessary in other functions. Does this mean that in the pursuit of becoming smarter, sacrificing other abilities like memory, emotion, and creativity is required? Before delving into this analysis, we must understand the true intentions behind creating these mini-brains. In the present era, medical science has advanced to the extent where any organ in our body can be kept alive outside our bodies and even transplanted into other patients. However, despite this progress, our brains remain the sole organ that cannot be transplanted, let alone kept alive outside our bodies. The main challenge lies in understanding precisely which part of the brain is responsible for keeping it alive. Even with advanced technologies like CT scans and MRIs, dissecting and experimenting on the brains of animals, even humans, has not provided a complete understanding. Therefore, in 2013, scientists at the Institute of Molecular Biotechnology in Austria conceived the idea of creating a brain from scratch to study it comprehensively. They extracted stem cells from the human body since stem cells have the unique ability to give birth to other cells. However, creating these cells requires growth factors, chemicals that transform stem cells into specific cell types, such as heart cells or brain cells. Among these growth factors is Noggin, which specifically transforms stem cells into brain cells. Injecting the Noggin growth factor into stem cells, the scientists placed them in conditions that closely mimic our body's conditions. Nutrients were supplied to these cells in a liquid form resembling blood, and they were kept in an environment replicating body temperature, similar to an incubator named an AVN. After waiting for just 3 to 4 days, the scientists observed a fascinating development. While many stem cells unfortunately perished, those that survived had successfully transformed into brain cells, and tiny brain tissues, akin to the tip of a pen, were formed. Encouraged by these positive results, the scientists allowed the brain cells to grow for about 60 days. When they re-examined the tissue, they noticed a significant increase in the size of these brain cells. Moreover, structures resembling the midbrain and forebrain had started to appear. At this point, scientists had triumphed in creating mini-brains in the lab, a feat that was thought to be unattainable before. To determine if these mini-brains could actually function, the scientists needed to translate their electrical impulses and establish communication with them. For this purpose, a microchip was designed, allowing the mini-brains to communicate through electrical impulses. Connecting this microchip to the mini-brains facilitated the translation of information into electrical impulses and the relay of signals back to the brain. Through this communication system, scientists initiated the training of these mini-brains. In the first step of training, they exposed the mini-brains to continuous electrical signals mimicking 8 different male voices for 2 days. This allowed the mini-brains to understand the differences in patterns between various voices. After this initial training, the scientists subjected the mini-brains to a test. They played 240 different audio clips of those 8 male voices and observed whether the mini-brains could accurately recognize and respond to these voices. Astonishingly, after just 2 days of training, the mini-brains exhibited almost 80% accuracy in identifying and reproducing the patterns associated with each voice, marking a significant success. Imagine yourself listening to songs by Arijit Singh, Sonu Nigam, or Lata Mangeshkar. Your brain swiftly matches the auditory patterns with its stored database to identify the singer's voice. Now, envision replicating this ability outside our brains, within AI-operated devices like Alexa or Siri. However, creating this capability in a lab-grown biological tissue was, until now, a concept straight out of science fiction. When scientists achieved this in lab-grown brains, it became evident that these mini-brains could understand and make decisions independently, potentially possessing intelligence and, perhaps, consciousness. As a science enthusiast, this achievement is significant. Presently, we have virtual reality cameras simulating our vision, microphones working like our ears, and touch sensors and mobile screens mimicking our tactile sensations. However, replicating our sense of smell and taste, managed predominantly by our brains, has been challenging. Some attempts have been made, such as the creation of an electronic tongue in 2011 that could detect specific chemicals and determine taste characteristics. Yet, it remains a formidable challenge because smell and taste are highly subjective. Some scientists have made attempts, such as the development of an electronic tongue capable of identifying specific tastes based on chemicals. For example, it could determine if a substance is sweet by detecting a higher glucose content or identify bitterness in the presence of nitrogen compounds. While this achievement is commendable, a fundamental question arises. Suppose someone has lost their sense of taste or smell, and we substitute it with an electronic tongue. In that case, can it accurately differentiate between the subtle nuances of, say, a gulab jamun and a rasgulla? The missing component here is a crucial one, a direct connection to our brain. If this electronic tongue could somehow establish a connection with our brain, it might significantly mimic our sensory organs, allowing devices to provide us with taste and smell experiences. Moreover, if we do not need an artificial nose or tongue and merely want to test a perfume online from the comfort of our homes, this technology could be simplified further. Imagine having a wireless system in your smartphone. You select a perfume online, receive an option on your screen saying, "Test it," click on it, place your smartphone on your forehead, and voila! The smartphone wirelessly transfers specific electrical impulses to your brain, allowing you to experience the fragrance in real life. This realization underscores the remarkable potential of our mini-brains. Just as the mini-brains recognized different voices with a microchip's help, we could potentially provide them training to recognize flavors and scents. This means that when brain cells equipped with a microchip communicate with electronic devices, these devices could function as our sensory organs, allowing us to experience the taste or smell of various things. Additionally, we could simplify this technology further for personal use. With this technology, we could develop devices like wireless chargers. Imagine a scenario where your smartphone is equipped with a wireless system, allowing you to select a perfume online, click Test It, and experience the fragrance through your smartphone by placing it on your forehead. This technology could be utilized even further. For instance, wireless chargers in the market already use magnets to wirelessly charge electronic devices. Essentially, they transfer electrical energy through touch. With further simplification, you could have your smartphone equipped with a wireless system. You select a perfume online, receive an option on your screen saying, "Test it," click on it, place your smartphone on your forehead, and the smartphone wirelessly transfers specific electrical impulses to your brain, enabling you to experience the fragrance in real life. This revolutionary technology holds immense potential. However, certain issues need to be addressed before it can become mainstream. As mentioned earlier, although these mini-brains are capable of thought, their thinking capacity is significantly limited due to their minuscule size, even less than that of lizard eggs. Moreover, experiments on these brains suggest that the microchip does most of the work, and without it, the mini-brains are merely a cluster of cells. Additionally, the microchip, being a machine, is prone to malfunctions. Instances like pacemakers malfunctioning, which regulate heartbeats through electrical signals, highlight the potential risks associated with relying on machines within our bodies. Pacemakers, for example, have been known to malfunction and inadvertently deliver harmful signals, posing severe risks to patients. This emphasizes that despite the trust placed in machines like pacemakers, they are still susceptible to technical issues. Moreover, having such machines within our bodies presents a dilemma. Imagine being aware that a machine inside you is causing discomfort, yet being powerless to do anything about it. Therefore, Calling these mini-brains perfect is far from reality, and mainstream application for users is contingent on the development of foolproof contingency plans. Nevertheless, scientists are undeterred by these challenges, recognizing the vast potential of this technology. Its medical applications, especially in treating various brain disorders, are immense. An intriguing experiment conducted in 2021 involved injecting lab-grown brain tissues into the brains of mice. Remarkably, after approximately 3 months, these artificial brain cells formed neural connections similar to those found in the natural brain cells of mice. This suggests that lab-grown artificial brain cells might integrate seamlessly with natural brain cells. The implications for treating conditions like Parkinson's disease, Alzheimer's disease, dementia, and other brain disorders, where natural brain cells are dying, are profound. Furthermore, since these brain cells are created using the individual's own cells, there is no concern about a cellular mismatch. In conclusion, this technology is not merely about integrating it into our touch screens, voice-activated devices, or enhancing our sensory experiences. It represents a new step toward the future, and the intriguing part is to witness how this technology will evolve and be utilized. Moreover, it raises questions about how we will employ and prevent the misuse of this technology when it becomes mainstreamed. I hope you found this video both informative and fascinating. If you did, please give it a thumbs up, share it with your friends, and don't forget to subscribe to our channel and hit the bell icon for more updates.